So what I'd like to do, though, is actually following on from Richard's introduction and words. I'd like to take us back to the 1970s, to my small laboratory at University College London, and give you some idea and some flavor of how we went about studying the hippocampus and how we discovered the place cells, and how we then took that idea and developed it into a much larger theory, which made lots of predictions, and which have then been tested over many, many years, both in our own laboratory and in many others throughout the world. And I'd like to then bring you up to where we think we are in the cognitive map theory of hippocampal function. And 5 or 6 years ago, I would have ended on a rather different note. I would have told you that I think we have all of the components, the bits and pieces that we need to construct the map. And that was primarily due to the findings in the Moser lab. And I'll tell you a bit about those. And then I'll tell you that we're not quite sure that that's correct. We're not out of a job yet. There are still things we have to learn about the components of the map, and I'll give you some idea of where things are going. So let me then start. Could I have the first slide, please? I guess I do the slides, yeah, okay. When, in the very early 1970s, I decided to tackle the functions of the hippocampus, it was primarily on the basis of work by Brendan Milner at Montreal, who had studied a patient called HM, who seemed to have, following bilateral hippocampal lesions for epileptic amelioration, recovered from the operation, had reduced epileptic seizures, but also had a dense amnesia. So at the time that I started to think about working in the hippocampus, it was very clear that the hippocampus had something to do with memory. But what that meant was of course, anybody's guess. So we decided to look at the hippocampus using microelectrodes in chronic, freely moving animals. At the time, and this is a wiring diagram of the visual system, which was produced by David Van Essen sometime later, but it represented people's idea about how you would go about studying a structure in deep structure in the brain, such as the hippocampus here. And the idea was, and this was promulgated by Vernon Mountcastle, Hubel and Wiesel, and many others, is that you worked your way in from the periphery, from the retinal ganglion cells, through the geniculate, into the early stages of the cortex, until you finally worked your way through this horrendous diagram, wiring diagram going through many, many stages and many, many interconnections until you came to the structure that you were interested in. So you looked at each stage at the transfer function, and then that would essentially take you all the ways into the brain and then back out again. Well, I thought, I was about 35 at the time, and I thought I'll be a very old man by the time I get through all of this to this point. So I thought I'm going to just go straight into the hippocampus. And most people thought that this was actually, to put it mildly, nuts. They thought that the idea that you would actually be able to make some sense of the activity of cells in an animal moving around an environment in an area that was very deep in the nervous system and very far away from the inputs and the outputs was just not going to be possible. On the other hand, I mean, I have a strong philosophical and psychological background, so my thinking was somewhat different. I thought, well, a lot of what we think is going on in the mind comes from what we call folk psychology. It's the story we tell ourselves about our own minds and other people's minds, and we have a whole set of notions about perceptions and memories and ideas and things like that. And I thought there was at least a possibility that things that we saw at the very far into the nervous system were actually coming closer to our folk psychological representations. It might turn out, and I think this is the case, it's certainly in the hippocampal system, it might turn out that it was easier to understand those than things at the periphery here, for example, in the retina where you were interacting with, the cells were interacting with photons and things like that. And that's what we did. So this is a picture of the hippocampus, and this is a cross-section through it. So myself and Jonathan Dostrovsky, who was a master's student at the time, decided to put microelectrodes into the CA1 field here of the hippocampus, allow the animal to recover, and then to and we had small, simple miniature microdrives to move the electrodes, these are extracellular recording electrodes, down to the CA1 field to look at the pyramidal cells and also the interneurons and see if we can make any sense of them. So here's the hippocampus in cross-section, a cartoon of it, and it's made up of the CA fields and the dentate gyrus. And then there are also these ancillary structures, which at the time we didn't know very much about and which we didn't actually think very much about, but have turned out to be very, very important as we learn more and more about how the overall system works. And they are the subiculus structures, the subiculum, presubiculum, parasubiculum, and the entorhinal cortex. And I won't go into any of the anatomy, but these are all very, very densely interconnected with each other. The entorhinal cortex projects into the hippocampus projects back to the spicula and then to the entorhinal cortex. So although many of us tried to make this into a linear structure, it turns out that it's much more reasonable to think of it as a kind of a set of interconnected structures and with lots of loops and feedbacks. So thinking that it was a memory system, Jonathan and I set out to do some very simple memory tasks. And one of the themes that I'm going to concentrate on in my lecture is the idea that we really need to have very good behavioral tasks if we're going to look at the function of cells in freely moving animals. And I'll tell you a couple of tasks that we and others have developed. So thinking that we wanted to do, to study memory, something akin to the sorts of structure, the sorts of things that HM, the Milner patient, had problem with, we developed several simple tasks. We trained the animal to press a lever to get food, to run towards a particular object to get food or water, and so on and so forth. Over a course of several months, Jonathan and I sat there, and in those days, we could record the cells on tape, but there was very little way in which we could correlate them with the animal's behavior. So we just sat there and watched the animal behaving and tried to make some sense of when the cells were fine. Luckily, in the hippocampus, as with many of these deep structures, many of the principal cells actually have almost no spontaneous activity. It's almost, I almost have developed what I call O'Keeffe's first law of the nervous system, which is that the important cells, especially deep in the brain, really don't have very much spontaneous activity. So you could sit there and look and listen to a cell for a long period of time and not even know you had a cell if you're extracellularly, recording extracellularly. But then every once in a while, the cell would go, brrr, and we would say, yeah, something's happened. So it's there, and it's responding to something that the animal is doing. It took a long time before we began to take a more ethological approach, an approach which said, okay, let's stop concentrating on memory, and let's just sit back and get the animal to do lots of things that it would normally do in its day-to-day life, eat, drink, sleep, and so on and so forth, and see if we can see any correlate of this burst of activity. And to make a long story short, what we found is that after looking at lots of cells over many, many months, I began to realize that the correlate of the cell firing was not what the animal was doing, whether it was doing a memory task or whether, and not why it was doing it, but it was actually related to where it was doing a particular behavior. So, for example, in this situation, the animal's running around on a tabletop, and the cell only fires when the animal goes over to A and B, as shown by the spikes here or these rate histograms. And it doesn't fire in other parts of it. And it turned out that the cell activity could be modulated a bit up or a bit down by other things the animal was doing in the place. And one particularly striking correlate was if the animal went to a place, say this location here, and expected to find food there or some other reward, and we had removed it. And then the animal would sniff around, and we found that that caused the maximal amount of firing. So on the basis of a small set of cells like this, each of which fired in a different location, I concluded that the hippocampus had something to do with providing a reference map of the environment. And there was already the idea out there, which had been generated by this man, E.C. Tolman, that as rats were learning mazes, the way they did it was not by learning simple routes, go left at the first corner, go right at the next corner, and so on and so forth. They didn't learn to make a stimulus-response set of connections. they had formed something that he called the cognitive map. He was rather vague about what that cognitive map was, but it was clearly the case that he thought that if an animal had a map like that, it would let it know the direction of the goal and how to get there. And he said things like, the animal has field map of the environment. It's not just connecting simple one-to-one switches between the stimuli and the outgoing responses. It's not a kind of a simple telephone circuit board. But rather the impulses, the nerve inputs, are actually worked over into a cognitive-like map of the environment. And this map provides routes and paths and so on and so forth. So this was the core of the idea. If there are a whole bunch of place representations, maybe they are forming the core of Tolman's cognitive map. Now, I don't think Tolman ever really believed that there would be a structure in the brain, and he was a psychologist, so he never looked at the brain, which actually contained his map. I think he thought it was the whole brain. And one of my jobs, I see it, is to sort of contain the map somewhere near the hippocampus and not let it sort of leak out to the rest of the cortex and the rest of the brain. Right, okay, well, if you want to sort of make this into a theory, then you better have a much better, clearer idea of what a map is. So around this time, and this was now in the mid-70s, Lynn Nadell came and joined our group on the top floor of the anatomy building at UCL, where Pat Wall was the head. And the two of us started to work on the overall theory as to what would it mean for a part of the brain to form a map-like representation? What else would you need besides these place cells? And what kind of predictions could you make derived from this idea that it was a mapping structure? So we said, well, it's obvious that you can, maps must be made up of these place representations, each representation referring to or representing a different location in a familiar environment. So you only need a small number of stimuli using triangulation. You can tell where you are by the doors there, the screens here, and so on and so forth. But that's not enough. Because to have a map, it's not enough to have an unorganized, unordered set of place representations. You need to know how those places are connected, how the representations of those places are connected together before you have a map. So we suggested that they probably were connected by distance and direction vectors, and I've represented that here in this vector diagram here, which connect the places in the map of the environment together, and that these vectors are created from the animal's own movement. And this is now an idea which is beginning to take hold in terms of general neocortical physiology, that any cell in the neocortex is actually getting several inputs, a bottom-up input, which represents the stimuli coming in from the outside world, and a top-down representation, which is actually making predictions about what stimuli will come in on the basis of the animal's movements or on the basis of other things. What you're seeing now might predict what you're going to see in a moment. So, and the animals could use this map to locate themselves in the environment and to move, and this is important, to move from one place to another by any available route. The map provides this kind of map, especially if it's working on some sort of vector computation of the sort that I'm outlined here, provides the animal with an incredibly flexible system for navigating. So if, for example, places are represented by firing in cells in the pyramidal cells A, B, and C, and they're connected by these vectors, then what you can do with a map-like structure like this is, if you're at A and you want to go to B in order to get, for example, a reward which you know is located at B, then the map should be able to generate this vector AB, send it to the motor system, and tell the motor system how to move in a particular direction for a particular distance to get to B. And then, of course, when you get to B, the sensory inputs that are coming there will actually confirm that that was a correct prediction. However, and importantly, if it turns out that that route is blocked for one reason or another, then the system should also be able to generate a large number of other vectors, for example, AC and CB, which get the animal to the same location. So it provides the basis for animals taking shortcuts, taking umwegs, and all of the kind of flexible navigational behaviors that we know that animals have. So on the basis of this, we analyzed a lot of the literature. We actually could show that we could explain a lot of the deficits that were seen after hippocampal lesions, which hitherto had been very difficult to actually to understand. So we made several predictions. And for example, we said, look, you don't have a map if you just have places. You need to have distances and directions. And so we predicted the existence of distances, directions, and also speed of movement. It's a no-brainer that you need to be able to take into account the animal's speed of movement as it goes from one place to another. If it moves faster, then it's going to go from one place to another faster. We also predicted that there would be deficits in place learning, the ability to know that the animal is in a particular location, navigation, and exploration. I won't go into why we predicted that latter. Okay, well, over the years, it turned out from work in our own laboratory and in that of many others, that we now know that in addition to the place cells, which are located primarily in the hippocampus proper, in the CA fields of the hippocampus, and which have the property of firing in one location in a reasonable size, 1 meter by 1 meter or 2 meter by 2 meter environment, but which, as shown in this polar plot here, don't particularly care about the heading direction that the animal is running in. So they don't care if he runs through the correct location going north or south or east or west. And it's actually that observation which first gave us the clue that these were not just simple sensory cells. A simple visual cell, for example, in V1, will fire when the animal goes through a particular location because that's the location where its receptive field is stimulated by a visual stimulus. But it won't do so if he goes in the opposite direction through the same place. These cells do in these open environments. In addition, we now know from work by Jim Ronk in New York that they're the obverse kind of cells. And these are the head direction cells, which Jim discovered in the mid-1980s. These are cells which fire if the animal is heading in a particular direction, anywhere in a familiar environment, but they don't particularly care where the animal is. So they're shown here, the cell fires, as shown in this false color plot here, all over the place. But it has a very nice preferred direction as shown by this polar plot. We also know that there are other cell types. For example, the grid cells were discovered by the Mosers, originally in the entorhinal cortex. And these are cells which fire not just in one localized place in a familiar environment, but in a whole series of localized places, as shown in this firing rate map here. And if you look at it, you can see that the firing locations are laid out on this beautiful grid pattern, where each of the peaks of the firing are located at the peak of triangles, if you will, or of hexagons. And it's that pattern which originally struck the Moses and which struck us as being a potential basis for providing the distance metric for the map. It's possible that the animal should be able to tell if this is a universal pattern, and as it's running in this direction through this grid field, it can just count the number of bursts of firing and get some idea of how far it's gone. And there are other cells which I won't go into, which give information about how far the animal is from the boundaries of an environment. So we think we have all the cells, although I will come back to the grid cells in the latter part of my talk, because we're kind of having a few worries about exactly what they're doing, and they're probably not, and I should flag this up, they're probably not giving the animal and the rest of the brain information about distances in any environment. Okay, now I want to spend a little bit of time on behavior. I know you're all physiologists, but I spend most of my time doing physiology, but I do think about behavior a lot. And it's very important that we have available to us, if we're going to record from animals that are actually moving around environments, or even if we're going to, as we do now, study the behavior of cells in head-fixed animals who are conscious but moving in virtual environments, we have to think very hard about the behavioral tasks. Because if you get the behavioral task wrong, you're going to ask the wrong questions, and the cells are not going to give you the right answers. So somewhere in the mid-70s, Richard Marius, who was a behavioral psychologist, came into our lab and said, look, you guys are serious, and that you think this part of the brain has such a specific function that if I make a lesion in it, the animal will no longer be able to navigate. I'm going to show you wrong. So he went away and he devised a task which was designed to test that prediction of the cognitive mapping theory. And it's a very, very simple idea. You place the animal into a bath of milky water so that it can't see anything below the surface of the water. And you let it swim around, and eventually it bumps into a hidden platform, climbs onto the platform, and says, I'm out of here. And you take them out of there, and you do this several times, 8 trials, 10 trials, and they very quickly learn that put down any place in that water bath, they will swim directly to the location of the hidden platform. And I'll tell you now, but I'll show you a picture of what it looks like from Richard's lab. It turns out that if you make hippocampal lesions, these animals are completely hopeless and helpless at solving that task. Okay? And the task really involves, since there is no information emerging from the platform itself, and you'll see in the video that you can show this, that it turns out that they are using the cues in the environment. They're saying, oh, there's a corridor over there and the door's over there and so on and so forth. And lots of experiments that we and many others have done have shown that that's exactly the basic set of information coming in that they use. So if I could run the video now, what you'll see is a couple of trials from Richard's lab. Here's the water maze. It really is just a rat swimming pool. And Richard was elected to the Royal Society a few years ago, and his sister, who has a bit of a sharp tongue, she's an architect, she said, did Richard really get elected to the Royal Society for inventing a rat swimming pool? And the answer is yes, not because it's such an elegant piece of apparatus, and I'll tell you why I don't think it's so elegant, but it actually pulls out in one simple behavioral task the essence of what the hippocampus is doing. So you can see, you start them from different ones. You can see here what happened, of course, is to make absolutely sure, since Richard's a psychologist, that the animal is not using the platform. He's pushed the platform way down so the animal can't actually stand on it. And you can see he swims around and around, and you can use a tracking system where you monitor the movement of the animal using an overhead video to tell where he is. Eventually, a clever rat will say, okay, I know that this is run by psychologists, so I'm going to get out of here. They've obviously removed the platform, and they'll eventually start swimming someplace else. Okay, so I'm a great fan of the water maze, and it has a huge number of citations. Every laboratory, every pharmaceutical company in the world has a water maze. And all you need is a little bit of plumbing and heating and Bob's your uncle. You can test any aspect of hippocampal function that you want to by just using the water maze task. So what do I like about it? Well, there certainly is no cue to guide the animal to the goal. He has to use some sort of triangulation of the room cues or some other distant information, which is just what the theory would like him to be using. They have to come at it from lots of different directions. So again, it can't be a simple sensory input. He can't just say, there's a cue over there, so I go in this direction. He's got to be able to do it heading in lots of different directions, which where he's facing in a different direction into the cue array. It's open, flexible paths to the goal, and it has there's a big effect of hippocampal lesions. It's what we call hippocampally sensitive task. So what's wrong with it? So I'm a terrible colleague to work with, and part of the reason that I've never had very many colleagues throughout my career is only partly because I never had any money. It's also partly I'm very, very critical. So when Richard first showed me this, I said, oh, Richard, come on, this is not a maze. Why do you call it a maze? This is not a proper maze. Mazes are situations in which the animal is faced with binary choices successively, and there are a whole bunch of properties which had been studied for many, many decades from around the 1920s to the 1950s in both the United States and Britain, about what you needed to have a proper maze. And the water maze really doesn't come up to the standards that one would like. And what I'm going to show you is I think we now have a proper maze, and I'll show you a bit about that. It's difficult to score performance. The animal is free to move in any direction. And so it's very hard, and what we tend to do is we tend to either take how long it takes him to reach the goal, that's assuming that he swims at the same speed all the time, or we take his heading direction every second or two. But neither of those is actually a very, very good measure, behavior measure. importantly, the choice at any point in the maze is not independent of prior choices. If he heads off in the wrong direction, then he's not going to sort of all of a sudden start going in the right direction. Importantly, and I'll show you this in a second, you can't use the water maze to record place cells because of the water environment. You don't have to be a crackerjack physiology to know that water and electricity just don't mix. And so if you were trying to record from an animal who's behaving, then the last thing you want them is to be swimming around in a water environment. And the other thing is, which is probably a little bit more, a little bit less obvious, is that in order to record place cells, you have to give the animal the opportunity to go to every part of the environment equally. So if I want to say that there's a cell that fires over here in the northeast part of the environment, then I have to give him a choice of going to the northeast, the northwest, and so on and so forth, or else I won't know if he doesn't go to some parts of the environment that the cell hasn't or hasn't fired. So over the years, I've devised a new task, and I'll just tell you about it briefly, and then I'll go back to the grid cells and tell you about some of our ideas about what the grids are doing, at least partly from some of our observations on this task. So what we've devised is a land-based water maze, essentially. It satisfies all of the criteria that the water maze satisfies and goes a long way beyond that. And what it is it's 37 in its present instantiation, 37 platforms, hexagonal platforms, each of which is sitting on a pneumatic tube, although more recently we're using linear motors, and each of which can be raised or lowered independently of the others. Okay, so when they're all up, then it forms a solid surface where the animal can run around just as it can in the water maze, or it could on the top of a tabletop. However, if we raise or lower only a small subset of the platforms, then what we can do is we can actually confine the animal's behavior to certain locations and give it only the choices that we want to give it at that location. So here's an example of 3 platforms raised. And if we have the animal on this one, then he's given a choice of going to either of those 2. So we can ask him to tell us that he knows where the goal is. He wants to go to the goal, but he's only given a very small, limited choice of platforms. So for example, we can put him on the blue platform here, tell him that or show him that the red platform is the goal, and then give him only the choice of A and B, one of which is more or less in the heading direction of the goal, whereas the other one is in a very different heading direction. And his choice here is obviously, the correct choice is to head towards the goal. On the other hand, we can also give him choices neither of which are heading towards the goal. So, for example, we can give him a choice between these 2 platforms if he's on blue. That is, he can either go in a direction which is about 90 degrees relative to the heading direction to the goal, or one which is virtually in the opposite direction. So what does he do in those situations? Can he solve that? And the answer he can, and I'll tell you what the implications are of that. So here's a short video clip showing you the behavior of the animal in this situation. The goal is down here, and he knows where the goal is. And we give him 2 platforms, one which is 180 degrees in the wrong direction, and one is 135 degrees. And what I like about this task is you can actually read the animal's mind. And that's what we want to do when we're recording from cells deep in the brain. We want to correlate not the firing of the cells with the animal's behavior, but what's going on in his mind. And so you can see he takes the choice here, which is 135 degrees. It's correct, but it's not something he really relishes. We can also force him to go someplace where he doesn't want to go at all by only giving him 1 platform. And he looks around, so he looks all around, and now he goes to the correct one because he wants to go down here. And here he's waiting, and again he goes here, and so on and so forth. And you can see he's actually making a very, very important decision at each point and telling us, and sometimes he looks and he says, whoa, wait a minute, I'm not so sure about that. Okay, what about, no, okay, I'll go here. And so on and so forth. So he's telling us by his behavior, and we can measure latencies, we can see how long it takes him to make the choices, and so on and so forth. He's telling us, and of course when he gets to the goal here, then he's fed at the end. So there's no food in the goals that he can smell until he gets there. So we can run lots and lots of trials of this and see how well the animals do, whether there's an effect of hippocampal lesions and everything. But most importantly, we can actually record from cells in the hippocampus and see what the correlates are. The animals learn this very, very rapidly. Here's 90%, reaching 90% in a very small number of days. This is 4 trials per day. That's not 4 choices. That's about an average of about 12, 14 choices per day. But they learned it very quickly. These are 2 groups, and they both learn it at the same rate, even though they're actually studied at very different times of the year. Now, there are several things. I won't go into this in any great detail, but there are some very nice things about this maze. One of it is that on the different choices the animal makes, that the animal makes, we can vary the difficulty of the choice. For example, we can vary the difficulty of the choice by increasing or decreasing the angle between the choices. So if we give him 2 platforms which are right next to each other, it's like a psychophysics test. He finds that harder than if we give him 2 that are far away. And it shows that the behavior is very good if the platforms are far apart and gets worse as they get closer together. And this is nice because we can actually vary the difficulty of the task as we go along. We can also vary we can also vary look at what the effect of the distance is from the goal. As choices are made farther and farther away from the goal, then the behavior also deteriorates. So that choices made from this ring here, which is only 1 platform away from the goal, are very good, whereas ones that are made from this outer ring are very bad. So that gives us control over it. Most importantly, and this is the real point, the behavior also and the performance also falls off as a function of the angle between the correct choice and the angle to the goal. So as you saw, the animal is quite capable of making a choice even when the correct choice of 2 is not taking him directly in the direction of the goal. He's heading in another direction, but it's still better than the alternative. And it falls off so that even when he's asked to make a choice which is greater than 90 degrees to the goal, he can still do it. Now this is important because even though the behavior, the performance is falling off, the actual performance, even at the worst choice, is much better than 50%. So that means that they can actually do the calculation. They can, in some sense, they can do the dot product or the inner product calculation where they take 2 angles between 2 platforms and they say this one is closer to the heading direction than this one. And we think this is a very important clue. We've always thought that the hippocampus might have something to do with vector computations of this sort. And I think this is the very first, albeit behavioral evidence, that they can do something like this. So not only can they calculate this vector and show that it's better than this vector, or calculate the sum of these 2 vectors is equal to this, but they can calculate all the other vectors and actually see which of those is better in terms of taking the animal more directly towards the goal. And of course, one of the things we're doing now is to look and see if we can see these signals in the hippocampus itself. We haven't seen them yet, but I'll just give you one hint of the way our thinking is going. There are lots of sinusoidal oscillations in the hippocampal system throughout. The theta oscillation, which in the rat runs about 8, 9, 10 hertz. And it's a very simple, there's a very simple theorem or analogy in engineering, which is phasor theory, which tells you how to go from vectors to sinusoids. So if you have sinusoids, you could, in theory, use sinusoids and phase differences in the amplitudes of the sinusoids to create vector representations and actually to do some of this vector calculation. Okay, well, we recorded from place cells in this situation, and one of the things we're interested in, although I'm not sure that I would make a great deal about the conclusions, is we're interested in whether the animal and the hippocampus sees the parts of the maze, small numbers of components of platforms together, as the same in some sense, and calling up the same representation as the entire maze. Okay, so does the animal think that when it's on the entire maze here are 37 platforms, and when it's on, say, 7 of those platforms together, does it think that this is part of this overall maze here? Or does it think it's something completely different? And the answer is that some cells in some animals think that it's the same, and some cells in other animals do what we call remapping. They actually change their firing location, preferred firing location. Here's one that keeps its firing location, showing that it is actually is locating the animal on the basis of distant cues, which don't change between these two conditions. So here's a cell which fires in the northeast corner, both at the beginning of a whole sequence of probes and at the end. And then when we give them subsets of the overall maze, we find that it only fires nicely in these two subsets here, which in fact correspond to this location. And it doesn't fire in these other ones. As I say, that's true of some cells, but not of the others. I won't show you ones which we map. We've also looked at these grid cells to see whether we see the same sort of thing, and we're finding the same kind of results. If we take a grid pattern like this, which fires with two firing fields on the east end here and say one here on the west, and then we break up the maze, we find that when he's in the west segment that you get one field, and when he's at the east segment, you get 2 fields. It's a little more difficult to show the correspondence with these multiple repeating firings, but you'll have to take my word that some of the cells do that. Okay, so we've also made lesions and shown, and here it's Ruth Wood is doing most of this work. If you make a big hole in the hippocampus using a neurotoxin, then you find that the animals are very, very poor at learning the maze. They do learn something, as people continue to point out. They're better than 50%, but they certainly are never, ever as good as the Interesting, if we now make lesions of the entorhinal cortex where the grid cells are, we get a much, much less effect. Much smaller effect. And in fact, this is not actually significantly significant between these two. It's almost significant. But the main thing that I want to emphasize is that the entorhinal cortex, which has the grid cells and which are supposed to be part of this overall circuit, doesn't seem to be producing such a great effect on, or be so heavily implicated in navigation. So what does that mean? And what that means is, I'll show you in a second. So here's where the grid cells are in the entorhinal cortex, and they have a very, very powerful, and most people think it's the main projection into the hippocampus, carrying that grid-like information and other kinds of information. Lesions here don't have as much effect, or hardly an effect on the navigation, whereas lesions here in the target in the hippocampus has a very profound effect. Okay, well, how do we explain this? The answer is, I don't think we can. But I'll tell you a few facts about the grid system, which I think is going to at least begin to get us to think in the right direction. So as you know, I showed you already, the grid cells were discovered in the Moses lab by Edvard and May-Britt, and they shared the Nobel Prize with me for that discovery, and it's a fantastic discovery. And anybody who sees this pattern, first of all, says, I just don't believe it. I don't believe that you can see a pattern like this in the nervous system, this incredibly repetitive, symmetrical, threefold symmetrical pattern, which you get when you record from animals as they're wandering around this environment in a helter-skelter way. You find that the cells only fire at the peaks here of these triangles, and that they have, you can characterize them in several different ways. You can characterize the distance between the peaks, and as I'll show you in the next slide, that changes depending on where you are in the entorhinal cortex. And they have an angle which they make with the walls of the environment. Well, when you see that, the Moses, we, and just about everybody else, and the computational neuroscientists went completely wild when this was presented to them. You're really hard put not to conclude that this is some kind of Cartesian metric system. This is the Cartesian metric for the hippocampal cognitive map. So I bought champagne for everybody, including the Moses. I thought it was a fantastic discovery. They've shown subsequently that, for example, that as you move down the entorhinal cortex here, the medial entorhinal cortex, the spacing between the grids at any given level changes, but it doesn't do so continuously. It does so in a quantized way, and for any given animal, there are about 4 different steps. So that if you look at cells up here in the dorsal part of the entorhinal cortex, you get a metric which is about 30, 40 centimeters between the peaks, whereas as you go down deeper and deeper, the spacing increases, and you end up with quite a large spacing when you get down, and nobody's really gone down to the very tip, but when you get down to this point here. Nobody has any idea why it's quantized. Some people think it's evidence that there are independent maps at the different levels. There is a bit of controversy as to what the scaling factor is. The Moses think it's about 1.45. We think it's about 1.65. It varies a bit, and there's a bit of a spread around it. But anyway, you can see why everybody thinks this could be the metric. Okay, so in the last couple of years, we've been asking the question, well, what do you really need for a metric? If you were going to have a metric for a mapping system, what properties do the cells which you are imputing that function to, what properties do they have to have? And of course, there have been a lot of thought about this by philosophers of science, people like Hans Reichenbach and colleagues. What do you need if you want to have a metric? And this is done mostly in the whole question of the philosophy of physics, but I think most of the arguments are pretty germane. So one of the things you need is you need a repeatable pattern. Something has to repeat. And I'm using this picture by Roy Lichtenstein here, where he lays these Bengay dots on all of his paintings. This is his trademark. But it turns out that these Bengay dots quite often are a grid pattern. And as you can see here with this six-fold symmetry. Threefold symmetry, sorry. And so you need to have something which is repeatable and hopefully periodic. I mean, a repeatable pattern would work as long as you don't want it to be a ratio metric. So you can have different kinds of metrics, and if you want it to be something like a ratio metric where the same distance occurs from here to here as occurs from here to here, then it should be some kind of periodic signal, and that's of course what we're seeing in the grid cells. It has to be internally consistent. So as you climb this mountain here, you don't want your metric to change. You want the metric to stay consistent despite the fact that you're moving around. As Reichenbach would have it, if you have a ruler and you move around the room and the ruler is expanding and contracting as you move, you just don't have a good measuring device. You can't use it. So it has to be the same going up. And finally, they have to be resilient. They have to be resilient to environments, different changes. If it snows, then one has to be able to sort of use the metric in any case. And we thought we had that with the grid cells. For example, the Moses showed if you double the size of the environment, you didn't change the grid structure. You just got lots more of the peaks. And even changing from light to dark, you got they still preserved the metric. Well, the first chink in the whole story came from work in our lab by Caswell Barry, who showed that not only are they not that the grid cells can be modified. So let me just put it directly, that the structure of the grids and the scale of the grids can be modified under certain environmental manipulations. And the obvious one is placing the animal into a novel environment. So if you put the animal into a familiar environment, record its grid structure, and then move the animal into a novel environment, the grids expand. And they expand differently for different grids. And then here's the experiment which shows that. So here are 5 trials. And here is the grid structure in each of these 5 trials. I won't talk about this. This is the autocorrelation, spatial autocorrelation, which makes it a little easier to see the symmetrical structure of the grids. Each of these boxes is the same dimensions, but they differ in terms of odor and texture and so on and so forth. So the animal has had a lot of experience with this box, which is then repeated in trial 5, but it has not had any experience previously with this set of boxes here, which is the same box. Place him in here, and you see the grid structure here, but when you move him to this novel environment, you can see it's pretty clear that the grid scale expands. And it continues to do so in the 3 repeats of that type of trial in that box, and then returns to the canonical shape when you put it back into the familiar environment. Over time, if you repeat this, even in the novel environment, the scale begins to return back to its canonical size. But for that temporary period in which the environment is novel, the scale expands. So that leaves you with the conundrum. Does the animal have a vertical scale? Does it know where it is in a novel environment? Or perhaps it just doesn't know. Perhaps animals can't tell how far apart things are in a novel environment. But it certainly started to raise concerns about the fact that the grids were some sort of a universal, fixed universal metric system. More worryingly, I'll skip that, more worryingly is work done in our laboratory by Julia Krupic and Marius Bauza, who showed that the walls of an environment have a very, very strong effect on the grids themselves. So if you do a simple thing like rotate a box, a square box, by 45 degrees here, what you find is that the grid structure rotates with the walls of the box. The grids are actually paying much more attention to the walls of the box than one would expect if there was some sort of a universal metric. Now, they have to know if it's going to be a stable grid structure, a stable scale across trials, then they have to somehow or other be fixed in some way to the environment. But this looked to us like it was a little bit too much of an influence of the walls. And the demonstration of the influence of the walls and how powerful it is and why these can't be a universal metric measuring system comes by recording animals and grids in non-symmetrical environments. It turned out until this experiment was done by Julia and Marius in last year that all the recordings from the grids were done in symmetrical environments. They were either squares or circles or such like. If you take a grid cell like this and you record in a square, then you get this beautiful grid-like structure with this beautiful symmetrical threefold symmetry shown in the spatial order correlatum here. If you now move the animal to a trapezoid type of environment, then the grid structure completely breaks down. And the easiest way to see this is if you look at this score here, 1.61, 1.41, that's a measure of the gridness. And something like about 0.3 is taken as anything above 0.3, which measures how strong this rotational symmetry is in the spatial order correlatum, is considered a very good score. If you put it in the trapezoid, it completely falls apart. And you can see that this very nice symmetrical structure here begins to be stretched out in one direction as opposed to the other. This has nothing to do with familiarity. The effect continues as shown in this line here over many, many days. The animal is very, very familiar with this environment. It doesn't actually have a good metric for that environment. We've done a little bit of work to figure out exactly what this is about. You can see here in another grid, what's happening is as the animal moves and as the grid structure develops towards the cone nose part of the trapezoid, then it gets bent and it begins to be elongated. And it's even the case that the fields in this end are actually slightly larger. You can't see it in this picture than in this end. And finally, what's happening is that in a square, the grid structure is the same on both sides of the square. And you can show that by just doing a simple correlation study. When you do the same thing on the trapezoid, so if this is the correlation or the similarity between two sides of the square, when you do the same thing in the trapezoid, what you find is that they're very, very dissimilar. And the grid score is very dissimilar between the left and the right part of the trapezoid, whereas it's very similar in this square. So something's happening in the trapezoid which is destroying the structure of the grid. And it's clear that what's happening is that the walls are no longer at right angles to each other, that the environment is no longer symmetrical. And this and other work that we're doing now and work that's coming out of other laboratories suggesting that the symmetry, this beautiful symmetry of the grids is actually being given by the structure of the environment. So that leaves us with a question as to, well, what is this grid structure telling the animal about the environment. Why is it useful, or under what circumstances would it be useful? And we think it's probably telling the animals something about the shape of the environment. It's known in humans, for example, that, and particularly about males who are navigating, that they use this shape of the environment to give them a strong clue as to which direction to go, and so on and so forth. Let me summarize then. The hippocampal formation provides a cognitive map, and I think that theory is still going and is still strong. And it can be used to identify locations and navigate flexibly, and I think flexible is the operant word here. And we're beginning, as I showed you, to get some idea of how you might create that flexible representation using vector type of calculations. The behavior in the honeycomb maze is actually, we think it's a very good platform for studying this. It shows that animals can actually calculate the direction to the goal. And even if they're not allowed to choose that direction, they can use that signal to decide which of the choices that it's given are actually the best. And navigation it's a hippocampally dependent task. We're still not sure why it's not sensitive to medial entorhinal cortex lesions. I think if we start to put walls into it, then I think we might find the things I've just showed you about what happens to the structure of the grids in walled environments, that it becomes much more important. We think that the grids, don't get me wrong, this is a fantastic discovery. They are a beautiful set of cells which have this fantastic firing patterns in certain environments, but not in others. There probably is some sort of an internal network. For many years, people considered various possible ways in which you could construct almost entirely by the internal network properties of the entorhinal cortex, this grid-like pattern. You can use continuous attractor network ideas. We've talked about interference between different frequencies of oscillators and so on and so forth. That can't be the whole truth. The environment has such a strong influence that it might turn out that it's the major influence, and that the structure that's created by the grid network itself is certainly modified and may be profoundly modified by the shape of the environment. So finally, let me thank the people who fund the work. The Gatsby Foundation, Wellcome Trust have provided major input to our laboratory, and they've combined to found this Sainsbury Wellcome Center for Neural Circuits and Behavior. And this is what it looks like, and we're building up the laboratories now. We have about half of the component of the labs have been filled, and we're still out there touting for talent. So if there's anyone out there who would like to come and study neural circuits at UCL, then they're welcome to apply. Thank you very much for your attention.